Tina to present their research on SARS-CoV-2. And the paper is called Data, Reagents, Assays, and Merits of Proteomics for SARS-CoV-2 Research and Testing. So Tina will give the introduction into the topic and explain you the proteomics workflows they have used. And Jana will go into detail. So for those who don't know Jana finished her PhD in December, sorry, and then basically jumped into the SARS-CoV-19 research. She did her PhD at the chair of Proteomics and Bioanalytics. She studied molecular biotechnology and did her bachelor's in nutritional science. I think her biggest achievement, I would say, was that she received the MCP Junior Early Investigator Award, and I think also she has a long list of honors at the awards, like the Maxwell Bilber Fellowship, the Karl von Frisch Award, and so on. So Jana is currently on hold. She wants to go to the US, but let's see if we have her still for a longer time around or if she can make the move at some point. So with that, I'm happy to share, give the word over to Tina.
Okay, can everybody hear me? Great. Super. Then I share my screen. So, as Karen already pointed out, this Advanced Mass Spectrometry seminar series is a graduate school event, and this is why one of our aims here in this seminar is also always to educate our participants a little bit in the basics and in the principles of mass spectrometry. And that's why we often have this educational teaser at the beginning of the seminar. And today it's my pleasure to give this educational teaser. And in this teaser, we usually cherry pick one topic, which is somehow related to the later research presentation. I picked today the topic of different MS data acquisition types used in proteomics. Yeah, therefore I will give you an introduction now into that, and I will already in that introduction try to highlight the differences and the advantages and disadvantages of these different data acquisition types, and already cross-reference to where later in the research or where we have used these acquisition types to study SARS-CoV-2, and Jana will then go much more into detail later on. Okay. So let's get started with an overview slide about the three major mass spectrometric data acquisition types that are currently used in proteomics. And these three different types are either data-dependent acquisition proteomics. This is what people also refer to as discovery-driven proteomics or shotgun proteomics. Then we have targeted data acquisition, which means targeted proteomics, or sometimes people also call it hypothesis-driven proteomics. And the third type is data-independent acquisition proteomics. And I will now go through these different acquisition types and once more very briefly explain you how the mass spectrometer works in these different types and for which type of question you would use which type of method. Okay, let's get started with discovery-driven proteomics, and because this is definitely still by far the most used method in proteomics that you will find in most proteomic publications, and it's historically also the oldest and most established workflow, I would say. Okay, so data-dependent acquisition proteomics you will always perform on so-called hybrid mass spectrometers. That means these experiments are done on mass spectrometers that have usually more than one mass analyzer. So the most frequently used setup at the moment would be so-called quadrupole Orbitrap or a quadrupole time-of-flight mass spectrometer. And these mass spectrometer in DDA, data-dependent acquisition mode, will always start their measurements with a precursor or MS1 full scan. And what that means is that whenever you have a mixture of peptides that are co-eluting at a certain time point X from your online HPLC, system that always sits in front of the mass spectrometer. So if you have a mixture of these peptides, they will all fly unaffected through this first quadrupole, and the Orbitrap or TOF mass analyzer will simply record all the peptide precursors that are eluting from your system at a certain time point. And these mass spectrometers are very fast and very smart, because what they can immediately do with these MS1 full scans is they can rank the detected M/Z values according to intensity. And then the instrument will switch to a second type of scan, which we call an MS2 scan or fragment ion scan, where now the quadrupole will get active, and the most intense precursor that was detected in the MS1 scan will be filtered in these first quadrupole. So in that example, only the green ball will be able to fly through the quadrupole, while all the other peptide balls will be filtered out. These green balls will then be fragmented in the collision cell, and the Orbitrap or TOF mass analyzer will record the fragment ion spectra, which is finally used to identify the sequence of that peptide. And as mass spectrometers are getting faster and better all the time, and they're actually fast enough not to just select the most intense signal, they can select also the second most intense, the third. And in these days on common mass spectrometers, usually people select maybe the top 20 to top 50 peptide precursors in one after another, and in a time frame of only a few seconds, 1 to 2 seconds. Once all these peptides are isolated and fragmented, the instrument will switch back to the MS1 scan and record the new peptides that will elute from the LC system a second or two later. And like this, the instrument will cycle through this MS1 followed by several MS2 measurement over and over again with a whole LC gradient. So in conclusion, this type of data acquisition, when would you use it? What's the ideal project to use it? And ideal projects for data-dependent acquisition are always projects where the aim is to map out as completely as possible the proteome content in one or in few samples. So if you want to identify in one or few samples as many peptides or proteins or post-translational modifications as possible, data-dependent acquisition is usually what people like to do. And another advantage of data-dependent acquisition is that you do not need to have prior knowledge to do the experiment, in a way that you do not need to know what are the biologically relevant proteins in my question, in my project. Just the opposite, data-dependent acquisition means discovery proteomics. So you actually want to figure out what are proteins that are relevant in a certain project. You do not need to know that upfront. And because of these two characteristics of data-dependent acquisition, we actually applied this method in our SARS-CoV research for two different sub-projects. So one thing that Jana will tell you later about that we did is we tried to map out as completely as possible the proteomes of cell line models that are typically used by virologists or by biologists to study viruses in general at the moment, especially SARS-CoV-2. So the purpose here really was to define the complete proteome in these cell lines and to give people an idea of which proteins are present in these cell lines and at which concentrations. And for that, data-dependent acquisition was the ideal method. And we also did another discovery experiment where the aim was to measure many proteins and find the ones that are interesting and changing. And this was an infection experiment where we treated one specific monkey cell line with SARS-CoV-2 virus. And we then wanted to compare the proteome before and after infection and look what are the proteins inside the host cell, inside the monkey cells that are changing, and thereby gain new knowledge about what's going on in these cells and how could the virus maybe be treated. Of course, there are also downsides of data-dependent acquisition. So DDA proteomics will be always biased towards the most intense or more intense peptides and proteins in the sample. It's because you always address a sample in terms of intensity from top to bottom. You always have this selection according to intensity. And that's why DDA will always be biased and much better detect this high intense one than low intense one. And this also leads to an intrinsic issue of technical missing values, so reproducibility between samples. So as the mass spectrometer by itself selects these most intense peptides, there is some stochastic process here, and you often do not get exactly same peptides identified if you measure the same sample twice in a row. So there's always technical missing values and reproducibility issues with this approach, especially for the lower signals, or mainly for the lower signals in the lower peptides in your samples. And that's why people thought about alternative methods to do proteomics. And one alternative one is targeted proteomics. And there are actually two different data acquisition types that fell under this term, and that's parallel reaction monitoring, PRM, as well as selected reaction monitoring or multiple reaction monitoring, SRM or MRM. SRM and MRM are always performed on triple quadruple mass spectrometers, while PRM is performed on usually quadruple Orbitrap or quadruple TOF instruments, so on the same instruments as also shotgun proteomics. And I would now like to focus on PRM only here in this example. So here you see how a PRM, what's happening inside the mass spectrometer in PRM mode. And on the first side, you might think, oh, wow, this looks super similar to DDA acquisition. You also have a full MS1 scan followed by a series of fragment ion scans. However, there are important differences between DDA and PRM. And the first important difference is that these MS2 scans that you perform in PRM mode are completely independent to the MS1 scan that has been performed initially. So these here, the peptide bolts are not selected according to what was detected in the MS1 scan. Instead, what you do in PRM is you tell the mass spectrometer in advance which masses it should isolate and fragment during data acquisition. And so what you simply do is you provide an Excel table where you give precursor masses of those peptides you would like to monitor. And ideally, you also provide retention time information. The cool advantage of this type of acquisition is that you will get very high quality and reproducible quantitative data for these peptides and these masses that you have selected here. But the downside is that this table that you can copy paste into your mass spectrometer cannot be endlessly long. So you are limited in the number of peptides you can monitor. Usually you can monitor routinely maybe in the range of 10 to 100 peptide precursors in one measurement. So in conclusion, PRM and as well as SRM or MRM, so these targeted proteomics data acquisition methods are ideal for projects that you already know which are the relevant target proteins or target peptides or translational modifications on peptides that are important for your specific project and your question. And it's the ideal method if you want to be really precise, accurate, and reproducible in terms of quantification, and also over many samples. So this is a major bottleneck or drawback of DDA is really that you have this issue with reproducibility over a lot of samples, and this is something that PRM can do much better for you. And PRM, and targeted proteomics in general, is also the approach that will give you maximal sensitivity. So you can span the largest quantification range with this technology. And because of all of these characteristics, we decided to use PRM in our SARS-CoV research for the diagnostics of a virus. So we performed an experiment where we targeted virus peptides directly in nasal swab samples from the clinic in order to see if we can diagnose virus infection with mass spectrometry. And for that, sensitivity and maximal accuracy and reproducibility of a lot of samples in a large clinical cohort was very, very important. The downside of SRM or PRM is that the number of peptides that you can target is limited. I told you this already. And the big downside is you need to know what to look for in advance. So you must have prior knowledge about which proteins to target, what are the best peptides for these proteins, what is the M over Z values of these peptides, what is the retention time of these peptides on your LC system. And in order to address this last limitation and to give to the community a resource that makes targeted analysis of SARS-CoV-2 proteins more easy, we performed another project in this paper where we used synthetic peptides of SARS-CoV-2 proteins to generate this prior knowledge. So to provide a resource where people can then just look up these coordinates and get a kickstart, and it's getting more easy to perform these targeted proteomic experiments. Okay, and just for completeness, I would also like to very briefly mention this third data acquisition type that people use in proteomics. We haven't used that for our SARS-CoV-2 study, but it's also the most recent and most novel data acquisition type that is used, but it's really currently gaining attention, and more people start using data independent acquisition. It's a very active research field. With this slide, I would just like to make the point that there is not just one data acquisition type that is used under this term DIA. Actually, there is a whole array of different, slightly different data, more or less different data independent acquisition methods that all vary in certain parameters, or they vary in how the data gets analyzed. However, I would just like to focus on one out of this array of methods, which is called SWATH-MS. And this is probably one of the most frequently used methods at the moment, one of the most popular DIA methods, but it's by far not the only method that is out there and that people use. So here we see what's happening inside the mass spectrometer in data independent acquisition mode. Again, on first sight, it looks very similar to DDA or PRM as well. You again have an MS1 scan followed by many MS2 scans, and the instrument always cycles through these MS1, many MS2s, MS1, many MS2s. The big difference of DIA compared to DDA is, again, the fact that all these MS2 scans that are performed are independent from what happened here inside the MS1 scan. So that's the same as in PRM as well, what I just explained to you. MS2 is independent from MS1, and that's also why data independent acquisition is called DIA. The second major difference of DIA compared to the other methods is the large precursor isolation window width that is used in DIA. So in PRM and DDA, you usually always try to isolate a single ball, a single peptide out of the mixture of peptides that coelutes. And therefore, you keep this quadruple isolation window width rather small, in the range of 1 m/z usually. In data independent acquisition, you open this quadruple window very wide. In the first publication by Gillet et al. Here in 2012, they had an precursor isolation window of 25 m/z. And that, of course, means that you no longer will have just one peptide isolated by the quadruple, but you can have many, hundreds of peptides co-isolated. They will all be co-fragmented, and the MS2 spectra that you will generate will be a strange, a very complicated mixture of fragments coming from all these different peptides. And what you do in DIA mode now is you stepwise use this precursor isolation window over a certain mass range, for example, from 400 to 1,200. And thereby you generate fragment ion information really from all peptides that are present in this mass range over the whole LC gradient. However, to make sense out of these complicated spectra was really difficult in the beginning. So you cannot use for DIA data the same data analysis tools as we use in DDA. And that was the novelty of this approach, how now to make sense out of that. And it's still a very active research field, and there are new papers coming out still all the time. I'm describing new ideas how you can make sense out of these spectra. Generally, there are two ways what people do. Either they analyze the data spectral library based. That means they use prior knowledge, as we do in PRM also. When you know how a peptide should fragment and where a peptide should elute, you can deconvolute the spectra, or people also start establishing spectral library-free data analysis workflows. In general, DIA is an ideal acquisition method for projects that involve not just a few targets, but really thousands of protein targets, where you still need precise, accurate, and reproducible quantification over large sample cohorts. But DIA is, in comparison to targeted proteomics like PRM, definitely less sensitive and less selective. And DIA, if you do the analysis in a spectral library-based way, requires these spectral libraries, and generating spectral libraries is work effective, and depending on which library you use, your data might change. So this is an additional hurdle that you have in DIA. But maybe these spectral library-free approaches that are emerging will also help to overcome this limitation in the future. Okay, with this, I'm at the end of my introduction into different acquisition mode. I just would like to wrap up once more these differences by using an analogy, which I think nicely highlights once more the differences between these methods. And this analogy is a sightseeing Munich. So let's assume we would like to visit Munich, and the data-dependent acquisition way of sightseeing Munich would be to take a look at the skyline of Munich and to pick the tallest building that sticks out of Munich in that skyline and to go and visit this building. And once you have visited that, you would jump to the second highest building, to the third highest building. And like this, you would make your way through Munich by always going one level deeper and deeper until you're at street level or until your time runs out. That would be a DDA approach to sightseeing Munich. And of course, you would get a nice overview over the city, but you also would maybe miss a lot of the very small but very nice things about Munich that are just on street level. The targeted proteomic way to sightsee Munich would be very different. You would, before you go into the city, first sit down at home and read a city guidebook. And based on what you read in that book, you would select, okay, what are the most interesting places for me personally? You would write them down. And once you're in the city, you would just go to these cherry-picked, pre-selected places. And these places can, of course, involve the tall buildings we have also seen by DDA, but they can also involve much smaller things that are, for you personally, maybe much more interesting than tall buildings. And the DIA approach, finally, for sightseeing Munich would be by far the most stressful approach, because your task will be to photograph everything about the city in a very strict and logical way. So you would be like a Google Street View car going around nonstop photographing everything. And only once you're back home, you would sit down, read the city guidebook, and based on what you read and what you find interesting, you would then look into your pictures, because you have taken pictures of everything. You can just pick out what you think is interesting and show that. So with this introduction and this analogy, at the end, are there maybe already questions to the different data acquisition types and why we use what approach for studying SARS-CoV-2 from the audience? Are there already any urgent questions?
So for questions, you can just unmute you or just use the chat version. However, just make yourself visible. So Tina, I think this was a perfect presentation.
I think so too. I hope you are well prepared now to understand everything about data acquisition that Jana will speak about now. Jana, the stage is yours.
Yeah, thank you. So I will share my screen. Okay, so yeah, this project was a joint effort from the chair of proteomics and BayBioMS here at the campus and the virology department of the University Hospital in Munich, and also JPT in Berlin was involved in this. And the whole thing actually started as an assessment of which COVID-19 proteins we can reproducible see in different kind of samples, but then it quickly morphed into a much bigger project. And in the end, we decided that we want to make all the data we acquired publicly available to facilitate SARS-CoV-2 research. And here's an overview of the different aspects we have investigated. And I want to start with the setup of a targeted assay, PRM assay for SARS-CoV-2 proteins, and then show how we applied that to the screening of patient samples. And then I will switch to two aspects where we use the hypothesis-free DDA approach. And one of these is the evaluation of the proteomes of certain cell line models that are commonly used in virus research in general, and specifically in SARS-CoV-2 research. And I will end my talk with showing some abundance changes of proteins, of host proteins in one of these cell lines after infection with SARS-CoV-2. So first of all, the question is, what do you need to do to set up a PRM assay? And first of all, you have to evaluate which peptides are actually, in theory, detectable by MS, and you do that by an in silico digest of the SARS-CoV-2 proteins. And by this, we ended up with 113 tryptic peptides, as you can see here. And these peptides were synthesized as isotopically labeled heavy peptides, and we pooled them and measured them in a DDA and a PRM approach. And by this, we were able to evaluate which of these theoretic peptides we can actually detect in the MS. And also, we generated a spectral library, which we then can use later for the final PRM assay. And we ended up with 98 SARS-CoV-2 peptides, which are detectable in the MS. So when you now want to set up a PRM assay and want to limit the time you spend on measuring all these samples, you want to further reduce the number of peptides that you actually target. And at best, you want to do that by depending on the abundance patterns of these peptides in an actual sample. So for this, we used a supernatant of a cell line that was infected with SARS-CoV-2, digested whatever was in that sample, spiked in our heavy labeled peptides, and measured them in a PRM mode. And then we looked for peptides that have a high abundance. So the higher the abundance, the MS2 signal, the better. And at the same time, we also prioritized for SARS-CoV-2 specific peptides. So in the end, we had a final PRM assay panel of 23 peptides that we wanted to target. And this assay we applied to patient samples. So we got 91 nasal swab samples of 2 different cohorts with different numbers of positive and negative cases. And these we also digested in gel. And as you can see here from the Comassi staining already, that the content of these samples is very different. So sometimes you have more protein in there, sometimes you have less protein in there. So the background is very different in these samples. And we also spiked in the heavy synthetic peptides to increase the certainty of actually identifying these peptides in the endogenous peptides in the patient samples. And then we measured everything in a targeted way using our built PRM assay. And we did that using a nanoflow system and a microflow system. And I just want to briefly point out the advantages and disadvantages of these two systems. So the first thing is that if you use a microflow system, you can run much steeper gradients and also decrease the time you need by decreasing, or the dead volume is not that important anymore. So you won't spend so much time on measuring basically nothing, because just the flow of your sample needs time to flow through your dead volume. But what is a known disadvantage of the microflow system, you lose sensitivity. And here you can see one of our example peptides of the spike protein and dilution row of this peptide. And you see that the signal in the microflow system is much lower compared to the nanoflow system. And usually you lose a factor of 10, at least for our two systems, nanoflow and microflow. But the advantage of a microflow system is that the retention time stability is much better. So again, you see increasing amounts of injected peptide quantity. And for the nanoflow system, here you see with higher loading amounts that actually the retention time is shifting. While for the microflow system, it remains pretty stable. And this is a big advantage if you want to set up a PRM assay where you, in an ideal case, want to have a very small retention time window that you specify in which the mass spectrometer should look out for this mass to fragment. So for the nanoflow system, we used 5 microliter of the initial patient samples, and that was actually already borderline with regard to overloading the system, so we simply couldn't load more. For the microflow system, we actually used the same amount as is currently used for the PCR analysis in the University Hospital in Munich, which is about 10 times more compared to what we used for the nanoflow system. And here you can see the results of our analysis. So the clinical samples are here ranked according to the virus load, according to the PCR result. And in red are samples marked that where we could detect SARS-CoV-2 proteins in our PRM assay. And you see that there seems to be a lower limit of 1 million genome equivalent.
Is it only for me? I think Jana got hold. Awesome. Maybe you can just switch computers, girls. Maybe Karen can go on the other side.
Can you hear me now?
Yes, we can
Okay.
hear you. Glad to have you back.
Yeah, technical issues, yay. Okay, so what was the last thing you heard?
I think it's, yeah, that we have this 1 million cutoff.
Okay. So yeah, we have this 1 million cutoff, but we have also these 2 outliers here, and we can't really say for sure whether these might actually be peptides that might map to coronaviruses, but not to SARS-CoV-2. So we couldn't disentangle that with certainty in this case. So, and here's again a summary of the results displayed as a visual contingency table. And you see that we don't have any false positives here, but as you have already seen in the last figure, we have quite some false negatives here, especially in the range of low virus loads. So this leads to a 100% specificity, but only 20% sensitivity of our PRM assay for SARS-CoV-2 detection in patient samples. And this is actually not so surprising because the high proteome complexity and the high dynamic range of these patient samples leads to a limited quantification range of the mass spectrometry readout. PCR-based methods, on the other hand, have the advantage of that they can amplify the analyte. And so we think that for large-scale testing, we still need PCR-based methods because they are much better with regard to sensitivity and probably also with regard to scalability. But still, this assay can be applied under other circumstances. For example, if you want to track the virus in a cell line using different drugs. And since cell lines are very often used in SARS-CoV-2 research, we also set out to characterize the protein expression profiles of 4 cell lines that are commonly used in virus research and in particular in SARS-CoV-2 research. And these are 2 lung cancer cell lines, CALU3 and A549, and for the latter one, we stably transfected them with ACE2, and this is a peptidase which is involved in the renin-angiotensin system, and has been reported to mediate entry of SARS-CoV-2, so basically functions as a receptor of SARS-CoV-2. And we also had 1 colorectal adenocarcinoma cell line, which is often used, and then 1 African green monkey renal cell line. And these apes are actually very often used in biomedical research. And this cell line particularly is used for production of virus and vaccines. And here for these 4 cell lines, we performed deep fractionation after protein digestion and measured 48 fractions for every cell line in a DDA-based approach. And then we identified about 10,000 proteins in each of these cell lines. And these data can now be used, for example, for cell type specific spectral libraries for DIA approaches, or also to set up PRM assays. But you can also use them to look for specific proteins where you might have a hypothesis why they could be interesting for SARS-CoV-2. And we did that with ACE2 that I was mentioning already, and another membrane protein, the protease TMPRSS2. And these have been reported to function as a combined system and mediate the entry of SARS-CoV-2 into host cells. And when you look here at these protein abundance distributions and where ACE2 and TMPRSS2 are located, it seems that these 2 proteins are mutual exclusive in our 4 cell lines, which is a bit surprising because you would expect them to be present, that both are present in the cell lines, especially for the lower ones here, since they are actually very permissive for SARS-CoV-2, so they have high replication rates of the virus. And so because we couldn't detect it in this DDA approach, but we know that the DDA approach can be biased towards high abundant proteins, we were wondering whether maybe we just don't detect it because it's lower abundant. So we set up another PRM assay for ACE2 and for TMPRSS2. And indeed, we could detect at much lower abundance ACE2 in CACO2 and CALO3 cells. And we couldn't detect it with Western blots, and we also couldn't see it in the DDA approach. For the other protein, TMPRSS2, we still couldn't detect it in ACE2 and VARO-A6, in ACE2, A549, and in VARO-A6 cells. So this might be a hint that actually there are other proteins involved in the entry of SARS-CoV-2, or maybe their presence is sufficient for the virus to enter the cells. And actually, there have been other proteins reported, and this is a very fast-moving field. So every couple of days, it seems there are new hypotheses what other proteins might be involved in the virus infection. And I just picked out 5 here, and when you now look where they are located in the protein abundance distribution, you will find, for example, that cathepsin B and cathepsin D in yellow and red are more commonly expressed across all these 4 cell lines. And this is actually also true if you look at different tissues, which you can look up at the in-house built database Proteomics DB. And interestingly, ACE2, which is like the most prominent candidate at the moment, is not expressed in lung tissue. So you at least cannot detect it. So if it's there, it is very low abundant. And so this shows that there is still much more research to be done, and the role of ACE2 needs to be clarified, and that we are still only in the beginning of understanding how this infection takes place in the human body. And to shed some more light on how a host cell is responding to virus infection, we performed another experiment where we used Vero-6 cells and infected them with 2 different doses of the virus for 24 hours. And we used Vero-6 cells because, as I was mentioning already, they are very permissive for the virus. So the virus is highly efficient in replicating in these cells. And we did the analysis in triplicates, and after lysis and protein digestion, we multiplexed the samples using tandem mass tags, so that we could pool the samples and then reduce measurement time by this. And here again, we did a deep fractionation with 48 fractions and measured the samples in a DDA hypothesis-free method. And here you can see how efficiently the virus proteins are produced in the host cell. So it's like more than 1.5% of the total protein content in the cell was virus proteins, and especially the nucleoprotein was very high abundant, and it actually ranked among the 30 most abundant proteins in the cell. We could also detect proteins which are not part of the final virus particle, like for example, this polyprotein R1AB. And this contains, for example, the polymerase, the RNA polymerase that is targeted by remdesivir. So actually the virus proteins were the proteins that showed the biggest fold change across the conditions, but we could also identify many host proteins that showed significant changes. So it was about 1,500, and we performed a cluster analysis with these significant proteins and could identify 6 main clusters, which are displayed here on the left-hand side. And then we performed a functional enrichment analysis to look for functional categories that are enriched within these clusters. And what was very apparent is that categories that are associated with membrane and extracellular proteins were enriched, particularly in the cluster that showed a downregulation upon the highest virus dose here. And on the right-hand side, you see proteins that are part of these enriched functional categories, and at the same time show an at least twofold regulation upon virus infection. And what you can see is that many of these proteins, of course, are also part of several of these functional categories. So as always with these functional enrichment analysis, there is quite some redundancy here. And on my last slide, I want to pick out some proteins and specifically show the abundance changes. As I said, we have many more. I just want to point out some of them. So for example, we saw many transcriptional regulators changing, and they were increasing and decreasing in abundance. For example, the transcriptional repressor MIR1 and the phosphatase RP-AP2 were among the most regulated proteins. And this phosphatase actually regulates the RNA polymerase, and by doing so, the transcription of small nuclear RNAs is changing, and by this also splicing changes. We could also see factors that are involved in innate immunity regulated, like for example, this protein, which is a growth factor and also is actually involved in the regulation of lysosomal function. And we did not only see them downregulated, but for some we actually saw also an upregulation, but only in the lower virus dose. And one example is this protease MASP1, which is an essential factor in the complement system. And this is quite interesting because it has been observed in COVID-19 patients that complement factors are going up. And while this reaction is important to fight pathogens, it can also harm connected with them. Okay. Okay, I'm back. So this protease here is important regulator of the complement system, and if that gets out of control, then host cells are damaged. And an overshooting immune response is actually one of the symptoms that has been observed in severe COVID-19 patients. And dexamethasone, for example, which is an immune suppressive, is now often given in the clinics to counteract that reaction. Then another thing I want to point
One second, we are trying to solve the problem. I think also your screen is stuck.
Yeah. So please,
you can just maybe go to my screen.
I think so too. Let's just switch quickly. Let Jana quickly go to your computer.
So this
works perfect.
last slide.
Yeah, so can you hear me now?
Yeah.
Last slide. I hope that's the last interruption.
You need to share the screen.
Mm-hmm. Okay, so I'm even connected with the LAN cable, but apparently, yeah, so proteases, I was talking about proteases. So MASP1 was not the only protease we saw regulated, but there were many more. And also proteins that were involved in cholesterol transport, and then phosphatidylcholine translocation, and also proteins that changed glycosylation of extracellular proteins. And this hints towards strong remodulation of the extracellular matrix. Yeah, so these were just a couple of examples. And another class we also saw was protease inhibitors. And this is also interesting because the virus basically needs active proteases to cut this polyprotein so that the virus replicase can be activated. And the second reason for why this might be interesting is that these protease inhibitors are often involved in regulation of coagulation. And blood clots is also one of the severe symptoms that have been observed in COVID-19 patients. And basically now doctors are discussing whether they should give even more of anticoagulants in the clinics, as they are anyway already like as a standard are giving for patients that are in intensive care units. Okay, so with this, I want to close my talk, and I'm happy to take questions.